My pleasure to introduce the next speaker for the second, Jeffrey Wong, who's from right here.
I'd like to organize a tour to Keio University and go into the human incubator and come out green and photosynthetic. So I would like to thank the organizers for this opportunity, and I'll describe our work on the synthetic genetic codes. All right, now here's the genetic code we're all familiar with, and the question is, why does it look like this? Very peculiar. Some amino acids have 6 codons, like leucine. Serine have 6 codons and 2 patches. Methionine, tryptophan has a single codon. It's not a kind of rational design a genetic engineer would sit down and draw up for 20 amino acids. So the question is why? And the best answer we have now is historical. And here we can see here's the aspartic biosynthetic family of amino acids. Aspartic gives rise to asparagine, gives rise to lysine, homoserine, methionine, threonine, and isoleucine. Here's aspartic, and here's asparagine, lysine, threonine, isoleucine, methionine. The whole family of these amino acids are sitting together in the same codon, adjacent codon domains. So this suggests then it's a family relationship. And here's cysteine and tryptophan are derived from serine. So this block used to belong to serine, and this becomes a contiguous serine domain, because as long as the same column or same row, that means only one codon, one code letter different. And also, this is selenocysteine. It's also derived from serine, and it also is given the codon UGA, sharing it with termination. Again, so these are sister amino acids derived from serine. So this is a historical code because of the biosynthesis bringing in new amino acids and causing a subdivision of the code. And so from this basis, it was proposed some years ago that we have phase 1 amino acids come from prebiotic synthesis, phase 2 from biosynthesis, phase 3 from post-translational modification, hydroxyproline, glycosyl asparagine, and so on and so forth. And this growth of the amino acid family represents what proteins want in natural selection. The more side chains there are, the more competent are the protein molecules themselves. And so this is how nature evolved, always looking for newer and newer side chains. So now there are more than 100 post-translational side chains. And so this is this theory was proposed 30 years ago, but has now been proven from the latest data from comparative genomics, because of the last 5-10 years, many genomes have been unearthed, and so I would refer you to this paper if you want to see the proof of this theory. Now, the problem then is facing us is the genetic code is shared by all the known living organisms. So that means this code, this universal code, predates the earliest branchings of the present-day living lineages, which is about 3 billion years ago. So this code has been frozen, unchanged, with respect to amino acids. Once in a while, the codon assignments do change, but the basic 20 or now 20 plus 2 amino acids never been changed. So frozen for 3 billion years. At the same time, the early evolution of the code suggests strongly that the early code had to evolve. So you have a demand for evolution early on, and the fact that it is frozen. So this has aroused a lot of questions, is the code a frozen accident? Can it be mutated at all? So this is, confronted with this question, we set out to test whether this code is in fact mutable. And the only way to do that is to try to mutate it. And by the way, the division of the 3 phases of amino acids, the phase 1 amino acids, the 10 of them are phase 1, and 10 of them are phase 2, based on the genetic code evolution. But now, the Kobayashi's group has produced 10 amino acids by high energy radiation on a slightly reductive atmosphere, and the amino acids they obtain are exactly the 10 phase 1 amino acids, and none of the phase 2 amino acids. Now, the latest work is this year from Pizzarello's analysis of one of the best preserved Antarctica meteorite, and she finds 10 amino acids on the meteorite. And those amino acids are exactly the same 10 phase 1 amino acids. So these 3 lines of evidence converge and tell us that the life started with 10 phase 1 amino acids. The others are latecomers, and they work their way into the genetic code. Okay, so the way they evolve is that an amino acid, typically, in some of the steps, it would go into the tRNA, and it is transformed into glutamine tRNA synthetase. And this glutamine sitting on a glutamyl tRNA would gain entrance into the genetic code, but it has acquired these codons. And the same aspartate can be transformed into asparagine on the tRNA, and serine into selenocysteine, into cysteine, and so on and so forth. And of course, the example of methionine into formylmethionine is one of the oldest examples. So this is how nature brings in new amino acids into the code. So we have then a RNA world scenario, the primitive genetic code with about 10 phase 1 amino acids, and they evolve and elaborate to the 20, and this gives rise to the last universal common ancestor. So if that's the case, how do we test the mutability of the code? We try to change the code by selecting bacteria to see how far we can change the code. And we know many amino acid analogs can be incorporated physically into proteins. But once you allow that to happen, the bacteria become unhappy, and they'll grow for a couple of generations, and they die off. So they cannot grow indefinitely. So they only incorporate the analog for the transitional period of a few generations. So what we want to look for is continual perpetual growth. And so we test the replacement of tryptophan by 4-fluorotryptophan and 5-fluorotryptophan and 6-fluorotryptophan. 5-fluoro is one of the most poisonous amino acids there is. From bacteria to mammalian cells, they all get inhibited by 5-fluoro. So we mutated bacteria, B. subtilis, and this is the wild type, QB928, and it grows very well in tryptophan, of course, and if you allow incorporation of phosphate into the agar, and there's a very hardly any incorporation in the presence of 4-fluorotryptophan. So the ratio is 730 to 1. And so if we mutate it, we get LC8, means a large colony on 4-fluorotryptophan, and then the ratio is 360. It's coming down. This strain, LC33 and 1.7. It grows as well, almost as well on 4-fluoro and as on tryptophan. Then we keep on going, and then HR7 prefers 4-fluoro. And HR15 far prefer 4-fluoro. It cannot really grow well at all on tryptophan. So it has lost the ability to grow on tryptophan. And so this is the chain of mutations. QB928 is converted to, in a couple isolation steps, LC33, it grows on 4FT, and as well as tryptophan, it grows LC62, 75, and then the HR7, HR15, it should be over here, and this one only on 4FT and not on tryptophan. So this one, again, this should be inside the box here, and these strains have lost the ability to grow on tryptophan. And then finally, we get LC88, which grows on, this is isolated by Dr. Flora Matt, and very patiently, she now gets mutants that grow on all three fluoroanalogs. And this is the results she obtained. And the wild type only on tryptophan and not on 4, 5, or 6 fluorotryptophan. LC33, 4 is okay, 5 and 6 are not okay. 62, 4 plus a bit of 6, and then HR23, only on 4, not on trip, very little on trip, and not on 5 or 6. And LC88, it can grow on trip or 4 F trip or 5 F trip or 6 F trip. So you can see that these experiments demonstrate to us that yes, in fact, in the earliest evolution, the amino acid code can change, and this gives the earliest organisms an opportunity to elaborate their side chains and select what kind of optimal balance they can have to work with. Because if you only give the bacteria only, say, alanine, valine, isoleucine, leucine, and no matter how long the proteins are, no matter how you mutate the sequence, the performance of such a proteome would be severely limited. So this is why the amino acid side chain is a critical attribute of how good the proteins are. And we can see from natural protein, from phase 1 to phase 2, and even today, so many post-translational modifications, phosphotyrosine, all kinds of modifications. The cells are always looking for newer and newer and newer side chains. And now, of course, in the 21st century, comes the synthetic biologists and the genetic engineers. They also want side chains. They want more and more proteins that break out of the restriction of the 20 encoded amino acids. So this way, we show in this example that yes, we can mutate the code and open up the code for evolution. And since that time, scientists in San Diego, in Schultz's laboratory, in Paris, so different parts of the world, in Germany, now there are now must be about 30 to 40 unnatural amino acids can be incorporated into bacteria for indefinite growth. So this way, the synthetically introduced amino acid side chains now already today outnumber the natural 20 amino acids. Because up to now, genetic engineers know very well you can take any protein and you can mutate the sequence at infinitum. And this way, you can try to look for the best sequence. But now with this kind of development, from now on, you can mutate not only the sequence, but increasingly the side chains that you want to grow the organisms in. So this way, you can move genomes around, mutate the sequences, and control what kind of side chains you can incorporate into the proteins. So now, here is the NMR showing the incorporation of the different fluoroanalogs. And here's of course, the next question then, these mutations, the whole series of mutations from the wild type to the different mutant phenotypes, where are the mutations? So this is now what is confronting us now. We have to figure out where are the mutations to understand what is the nature of the mutations inside the B subtilis that allows us, the cells, to work with new genetic codes. So this way, that will help us understand what is happening and also help us in the future how to introduce changes into the proteome. And here's some of the mutants that Flora found in some of the proteins. Here's a wild type mutant MR3, and then you have a glycine mutating to a serine, and here's a wild type mutant. The thinking is that a tryptophan residue, for example, let's say in the wild type, many residues of tryptophan do not really care if you change it to 4-fluoro, because the context in that residue allows the replacement without too much disturbance of the function. But there are some critical residues which can only work with tryptophan. So under these circumstances, you put in 4-fluoro into this place, or 4 or 5-fluoro, and this protein will not function anymore, so the cells will die. So what happens under these circumstances, if there is a mutation nearby this residue, and this mutated from H to J, then this changed microenvironment around the TRP residue now would allow the replacement of TRP by 4-fluoro, and the protein function in either case. This way, then this will be able to work and maintain, sustain growth and reproduction. And then next step, if this residue can work with either TRP or 4-fluoro, and if this amino acid mutates further to another residue, changing the context so that tryptophan can no longer function. It can only function with 4-fluoro, then this mutant can no longer grow on tryptophan. So this is the mechanism we believe is happening to bring about this kind of selection of amino acid for the proteome. And as I mentioned, in the isolation, we only use a few isolation steps to change the proteome identity of the encoded amino acid. And this was really quite surprising. That tells us that not too many critical residues in the proteome of B subtilis really decides the rejection or acceptance of tryptophan versus 4-fluorotryptophan. And to look for more evidence, here we grow the organism. We have a complete growth medium to begin with, but we can withdraw one nutrient at a time. We can withdraw A, G, UCT, thiamine, niacinamide, pyridoxal, and so on and so forth, and see what happens. And if the cells are growing on 4-fluorotryptophan, you withdraw each of these nutrients, the growth is not very much affected. That means all the enzymes in the biosynthetic pathways of these nutrients, they can accept a change from tryptophan to 4-fluoro. And the same with 5-fluoro, 6-fluoro. The majority of the pathway enzymes do not get upset when you replace the amino acid residue. However, if you withdraw threonine here, then the threonine biosynthetic enzymes apparently do not get very happy. You start to lose growth. Leucine worse, and the thionine even worse. So here, you withdraw these nutrients, and you see increasingly, there's a loss of function of cell growth. And the same here. So out of the many pathway enzymes, biosynthetic pathway enzymes, only a few of them are really ultra-sensitive to a switch of the amino acid side chain. And it is these resistant amino acid residues that has kept really the genetic code from unchanging, from the invariant state for 3 billion years. These residues maintain the collection of the 20 amino acids that we have today. So this then really confirms the isolation procedure that you can get a genetic code mutation only with a few mutational steps. So here's some of these other enzymes which seem to have show some kind of change, a residue from one strain to another. This is a DNA A, isoleucine S, dihydrolipoamide acid transferase, and these are two proteins of unknown functions. So now what we have to do is to identify these proteins and test the function one by one to see whether this particular protein does in fact lose the function when you mutate from G to S. So this then will be the next phase that has to be done, is to really pin down the positions of the mutations. As you can see here, this is the strain HR23. It cannot grow on tryptophan, but it can grow on 4-fluoro. So when the cells are growing on 4-fluorotryptophan, you put the tryptophan in the hole. This is a standard antibiotic assay. You put the inhibitor in the center hole and see what happens. Of course, it causes inhibition. So tryptophan is an inhibitory analog. It used to be one of the 20 canonical amino acids, has now become an inhibitory analog against these cells. So you can see this zone of clearing where the cells fail to grow because of this tryptophan diffusing out. But in this clearing zone, you can see some mutant colonies, and when flora grew it up, this is one of the resistant colonies, and this one no longer sensitive to inhibition by tryptophan, because this strain now can grow on either tryptophan or 4-fluorotryptophan. So this then is what this work then is established that the alphabet can change. And in this article on New Scientist, the author says, poor, pitiful evolution and proteins constructed from just 20 amino acids and DNA stitched together from an alphabet of 4 measly chemical letters. And say goodbye to evolution. It's time for a revolution. And this article, the name of the article is called Life, the Sequel, because the life for the past 3 billion years is the life based on a proteome of 20 amino acids. But from now on, life, the sequel, is sort of unlimited amino acid side chains. 1 minute. And then the other one, in this article, it suggests that HR15 is not only a picky eater, but an entirely new type of life, because the genetic code, as we know it, the 20 amino acids, is a fundamental characteristic of life on Earth. The kind of life we know, ourselves, the bacteria, the viruses, they all use these 20 amino acids. So when you change the proteome building blocks, then you are now, according to this article, you're looking into a new kind of life. So at least we have a first example of a synthetic cellular life. And I would like to thank Hannah Xu, Flora Matt, Shu-King Ng, and Antoine Danchen, Andy Ellington, and Huai-Ming Yang for their collaboration. And right now, Andy Ellington's group has extended our approach to E. coli. So they have isolated strains which they call uncoli. And they use the unnatural amino acids instead of the natural amino acid tryptophan. And Antoine's group and Huai-Ming's group are now working with us to try to pinpoint where are the mutations in the genome that of B. subtilis, whereby you change it, and through it you can change the genetic code. Thank you very much.
Okay, thank you very much. We have time for one or two questions. Yes.
Unnatural amino acid. PDS train.
How long, Flora? Maybe 3 years. 3 years. Very patient. There must be some faster way in the future. So you look for minimal growth. You have a gradient of inhibitor. You see the cells adapting. And then gradually isolate them. Especially the 5-fluorotryptophan. She spent a lot of time, a whole year, isolating that strain because it's really a very toxic poison.
Right. So you're using growth as your ultimate selection.
Continued growth.
Continued growth.
Yes.
But can you use this kind of approach to select for new functions as well? So you don't have that same, you have a requirement for growth, but also you want to change proteins so they acquire new functions.
The functions more or less you have some idea what new functions are by the kind of amino acid you use. For example, with 4-fluorotryptophan is very interesting. It does not fluoresce. Tryptophan fluoresce. 5 and 6-fluoro, they're all strong.
What I'm saying is if you change the structure of a protein because you put a tryptophan in, can you acquire new functions?
Yes, not new function,
for
new
example, drug resistance.
New properties. In the case it loses fluorescence. But in the Schultz's group, they put in a new amino acid which is susceptible to cross-linking. Then they take the proteins out and they zap it, say photoactivate, it becomes cross-linked, becomes very stable. So in other words, you can use analogs or choose analogs whereby you have some idea what kind of functions it may give you.
Okay. One last question.
You mentioned that more than 30 unnatural amino acid has been introduced into the bacteria. And is that it must be encoded by a special codon like the stop codon, or it is that you could encode it by any codon you like?
Good question. There are two ways now to change this amino acid protein and proteome. The way I've just described is called the top-down. You're changing every residue in the whole proteome. But some years ago, we found that you take the synthetase and tRNA from 3 domains. The archaea, bacteria, sometimes they do not cross-charge. So if you have, and this finding has been made use of other groups you put in synthetase and tRNA, they only recognize one another. So they do not recognize the endogenous tRNA and synthetase inside the cell. So you have an orthologous tRNA and synthetase pair that can only cross react. So you choose a rarely used codon and you slip this orthologous tRNA into that codon. In this way, you can choose certain residues, predesignated residues designed by you, where you want to put the unnatural amino acid. So you have a top-down approach and you have a designated approach, and I'm sure in the future people will learn how to use combine on other approaches so that to steer them into, there are some codons which are very rarely used and sometimes hardly used at all. So this way will be open to the genetic engineer to move that codon into any protein and put into a designated amino acid.
Okay, let's thank our speaker again.